Hello, my name is Keith Gardner and I'm from NIAB in Cambridge in the UK. Thank you for inviting me to present my work at BGRI. Today I'm going to introduce a collaborative project I am leading on adult plant resistance to wheat striped rust, involving NIAB and 7 North European breeding companies. The project is called Yellowhammer, and the aim is to improve breeding for durable striped rust resistance by determining the most effective combinations of adult plant resistance loci for stacking in breeding programs. As is widely known, wheat striped rust is a major concern for European wheat growers, especially with the incursion of a novel, highly diverse pathogen population in the last decade, and the subsequent dramatic changes in patterns of resistance which have been occurring as this pathogen population evolves. Underlying the project is the idea that using multiple race non-specific adult plant resistance loci is an effective long-term control strategy. In previous work with this consortium, we use genome-wide association mapping to identify many useful resistance loci segregating in present-day and historic germplasm, but also observe that these loci interact in complex ways, such that finding the most effective combinations remains a challenge. In this project, we aim to identify further resistance loci, phenotypically and genetically characterize the most useful loci, determine how these loci interact together, and identify the optimal combinations to maximize striped rust resistance and resistance durability. Here I show a project overview. We have 2 germplasm development components shown in yellow, feeding into 3 experimental components in light orange, with outcomes shown in dark orange at the bottom. And I'll go through each of these in a bit more detail. Firstly, we have a new 400+ line GWAS panel with a wide diversity of Northern European lines. And we've also developed more than 20 biparental crosses involving parents which are highly resistant, but not carrying any major resistant loci which we could detect in a GWAS panel. Most of the biparental population show very promising segregation patterns, as can be seen here. In combination, these resources are being used for genetic analysis of interactions and new resistance locus discovery. But in addition, they also enable us to annually monitor changes in the pathogen population as known resistance loci become ineffective or new loci emerge as effective. Our second germplasm development component is development of nil pairs for the most important resistance loci, which differ only at that resistance locus. In some cases, we've developed double nil pairs segregating for 2 resistance loci, as seen here on the right, enabling precise characterization of potential interactions between the loci. Development of nil pairs has been greatly facilitated by the collaborative nature of this project, enabling screening of a very large number of crosses across all the company breeding programs for potential nils. Extensive field trials are at the heart of this project. The GWAS panel is being grown at 14 sites per year across 4 years of the project, with an additional preliminary year of data for a subset of sites. Biparental populations and nil pairs are initially grown at a single location and subsequently distributed more widely. Overall, the field work is enabling us to evaluate the most effective allele combinations against a wide diversity of pathotypes under a range of different abiotic conditions. Further field work will entail the use of nil pair crosses to investigate resistance expression in heterozygotes, with hybrid wheat breeding in mind, and towards the end of the project, a cross-company meta-analysis of the pleiotropic effects of resistance loci on yield. To investigate the underlying mechanisms of resistance in more detail, we are carrying out extensive microphenotyping of the plant response to inoculation with different pathogen races to evaluate how, when, and where the resistance response occurs. For example, is it a hypersensitive response, indicative of variation at a major R locus in the plant immune system, or a more subtle response? Both nil pairs and a wide range of wheat varieties are being tested using this approach. In the third experimental component of the project, we are conducting differential expression analysis on the nil pairs in response to pathogen inoculation to understand more about the gene regulatory networks underlying the resistance response and how these relate to the best combinations of resistance alleles. So overall, the project systematically addresses the overarching question: what is the optimum allele stacking strategy? Is it better to combine loci that interact additively or loci that interact synergistically, or some combination of the both? Is it better to use loci that are part of the same gene regulatory network or different gene regulatory networks? Or loci that act at different times and locations in the plant or at the same time and location? Or more generally, what is the optimum mix of major resistance loci and weaker APR genes? More immediately and practically, the aims of the project are to provide new resistance sources, markers, and knowledge to help European breeders produce more resilient wheat varieties, equipped to resist existing and potential new strike rust races. Such varieties will be less likely to suddenly fail due to strike rust outbreaks, and therefore likely to last longer as viable planting options, providing stability benefits to both farmers and at later stages in the wheat supply chain. By increasing knowledge of variety-specific timing of resistance, we also hope to aid farmers optimize pesticide application, potentially ensuring reduced fungicide usage. I will now briefly present a few results from the project so far, focused on the field component. The European strike rust situation has been rapidly changing since the 2011 incursion of the novel pathogen population. Aside from the initial changes, there have been several subsequent years in which major changes in resistance ratings of varieties have occurred, correlated with changes in the pathogen population. For example, 2016 saw several UK recommended list lines simultaneously losing their resistance. Our GWAS panel results over several years have clearly shown the patterns of changes in pathogenicity, while highlighting the specific resistance loci involved in the change. Here I show results from a subset of roughly equal disease pressure trials in 2012 to 2014 and 2018 to 2020. The dates are shown at the bottom of the figure. Each row represents a single important resistance hit of interest, and the bars indicate the excess percentage of strike rust found on susceptible compared to resistant lines in a particular trial. The scale bar runs from 0 to 40%. Green coloured results are from UK trials, blue from non-UK, with only a single representative measurement date shown per trial. On line 1, you can see a typical resistance locus that was widely effective prior to the 2011 incursion, but shortly thereafter was overcome. In contrast, on the third line, you can see a locus that had no effect on resistance prior to the incursion, but has subsequently been widely and highly effective and continues to be so. On the other hand, on the second line, hit B, was partially effective before the incursion, became very important in almost all trials after the incursion, but has just been overcome in 2020, where it no longer offers resistance in any trials. This will result in significant changes to varietal rust ratings for several varieties in the coming year. The fourth line, hit D, represents a new hit since 2016, which may be increasing in usefulness as time progresses, whereas hit E represents a double change. Prior to the 2011 incursion, this hit provided good resistance, after which it became initially weaker, usually being effective only in combination with other resistance loci. But in recent years, the alleles have essentially flipped. The previous susceptible allele is still susceptible, but the former resistant allele is now hypersusceptible. Finally, hit F on the bottom row is representative of several hits which provided weak or occasional resistance before the 2011 incursion and continue to do so now, being potentially weak but useful loci for stacking, depending on their interactions with other loci. Moving on to interactions, we are still in the process of comparing approaches for analysing the large number of field data sets for genetic interactions between markers. In preliminary work, we found a wide range of interactions are observed. These include additive interactions, shown in yellow here, where the presence of the resistant allele at both loci, on the left, is roughly the same as adding together the effect of the two single loci. We also see positive synergistic interactions, shown in green, where the presence of both resistant alleles results in much greater resistance than the sum of their individual effects. And redundant interactions in blue, where each individual effect is generally so strong that combining them offers no extra resistance benefits, although to a breeder, this may offer insurance against loss of a resistance allele. Finally, we also find suppression interactions in red, whereby the wrong allele at a modifier locus can completely negate the effect of a resistance allele, despite not being detrimental in the presence of the alternative susceptible allele. The table shows results from a student PhD project characterizing the frequency of these different patterns of interaction in disease trials of Niam's magic population. The take-home message is that all patterns were observed, but the synergistic and redundancy patterns were the most common types. Finally, this project is a large collaboration to which many people have contributed and continue to contribute, and I would like to acknowledge all of them here, as well as our funding providers, BBSRC and AHDB.